Today we're going to be talking about the process of gram staining. Gram staining is a technique that we use, especially in microbiology, to identify bacteria. When you're looking at a bacterium under a microscope, it's not always easy to tell what you're looking at. They're incredibly small, and even using a highly advanced microscope, they don't have a lot of defining features between them because they're just single-celled organisms. So gram staining is something that we use to separate bacteria into one of two groups. They're either going to be gram positive or gram negative. And the way that we're going to be able to separate these bacteria are based on their cell walls. So a little bit different structure within their cell walls, and so that allows us to separate most bacteria into one of these two groups, which we're going to kind of allow us to differentiate all the different bacteria that we have. So a little bit about cell wall and how we're able to do this. So we're going to have a little bit of a refresher here. The cell wall of a bacteria, as we know, gives a cell shape, gives it protection, and it prevents the cell from becoming a hypertonic environment. So in a hypertonic environment, that makes a cell lose water. So this is what would happen if we take a cell and we put it in a solution of really salty water, right? The water outside of the cell wall is the salt outside of that cell wall is going to draw the water inside the cell out, and it's going to cause that cell to shrivel. This is why we don't water plants with salt water, right? We give them fresh water so that the water moves into the cell, not out of the cell. So the cell wall protects that and makes it so that the environment inside the cell is not hypertonic. So severe water loss inhibits them from reproducing, which is why, like I said, salt can be used to preserve food, because if we put enough salt in food, then the bacteria won't reproduce, and this will prevent food from spoiling. So it's one of the reasons why we use salt, because it basically damages the bacterium and kills them so that we don't have to worry about our food going bad. So that's all important because we're going to use that principle of that hypertonic environment to help us through this gram staining procedure. So what we're going to do is, to start, after a bacteria is fixed onto a slide, okay, we are going to put crystal violet on that slide, which is a dye that we use commonly in microbiology. Okay, that crystal violet is then going to be absorbed into the cell wall, and it's going to link with the peptidoglycan, which is in the cell wall. Okay, we are then going to add iodine. Again, another stain that we use. It's very commonly used to help us identify different starches and things like that. So we're going to add iodine after that, and that's going to bind with the crystal violet to make it insoluble to water, which is important because we don't want to be able to just add water and wash all this away. So the iodine is then going to bind with the crystal violet, and that makes it so it won't dissolve. Then we're going to take that slide and we're going to rinse it with alcohol. This is why we need it to be insoluble. We're going to rinse it with alcohol, which is a decolorizer, right? The alcohol is going to take the color away. This is where our hypertonic environment comes into play. Once we add the alcohol, that is going to shrink the cell wall, and it's going to dehydrate it, okay? And the crystal violet and iodine complex in a gram-positive bacteria, it gets stuck inside the wall. And the gram-negative bacteria is going to lose all color and become colorless. Okay, so once we take that alcohol and we rinse the slide off right over the bacteria, if it's gram-positive, it's going to be stuck, and we're going to have the color is going to be stuck there. If it's gram-negative, then all the color is going to be washed away. But we're not done there. Okay, saffron is a red-colored dye. It's added, and it's going to be absorbed by the gram-negative bacteria to make them visible again, because we don't want them invisible, because then we're like, did we wash it off the slide? Is it still there? Are we looking at nothing? Gram-positive bacteria, what separates them from gram-negative bacteria is they have a simpler cell wall. Okay? It is a thick layer of peptidoglycan, which makes them stain darker. So when we go through this process, okay, what we're going to see is they're going to show up purple. So we added the crystal violet, right, which is kind of a purple. Then we added the red, and then it's going to absorb all that color. And so when you're looking at a gram-positive bacteria under a microscope, they are going to appear a purplish violet color. Okay, so they are typically a less threatening bacteria species, meaning they don't cause as much trouble for us as humans as a pathogen, simply because They have a simpler cell wall, and most of our antibiotics work to break down cell walls of bacteria. So these are the type of bacteria that we can treat pretty easily with an antibiotic. It's going to destroy that simple version of the cell wall, and then once the cell wall is depleted, the cell itself is dead and it no longer can hurt us. So we don't worry as much about gram-positive bacteria. It doesn't mean they can't cause problems for us. It just means that they're a lot more treatable with the antibiotics and things that we typically use to treat bacteria. And again, under a microscope, they are going to have a purple appearance because they're going to absorb all of that color that we went through in the staining process, okay? Sometimes though in these gram-positive bacteria, they're not completely harmless. They may have a lipid membrane that is often toxic. And that protects them from the body's defenses, and it keeps antibiotics from entering. So it's not all, it's not all a good thing. Many antibiotics, like I said, are going to target the cell's peptidoglycan and stop it from cross-linking, which destroys the cell, and that allows them to effectively kill the bacteria. But they still can have a little bit of that toxicity in the membrane. And kind of add an extra layer, so they may be a little bit more stubborn, and they may prevent those antibiotics, but on the whole, they're a lot safer for us than most of our gram-negative bacteria. The gram-negative bacteria, they have less peptidoglycan in their cell wall. That's why they stain the lighter color. Okay? They're going to show up pink under a microscope after they're stained. So gram-positive, purple; gram-negative, pink. Gram-negative bacteria tend to be more threatening as a bacteria. They are increasingly resistant to more or most antibiotics because, again, most antibiotics are going to work on targeting that peptidoglycan, and these bacteria don't have any. So when the target that you're looking for isn't there, it renders most of your treatments ineffective. Okay, so these guys a little bit more stubborn, a little bit, takes a little bit more work to rid our bodies of these types of things. So that's why, like I said, we worry about them a little bit more. Again, if we are going to, if we don't know what is wrong with you, okay, this is where this comes in handy. We're going to take a sample of cells, we're going to stain it, and we're going to look at it in the lab and say, oh, it's purple, it's gram-positive. All right, let's treat it with this. If we take that sample from you, we take a culture, we stain it, we look at it in the lab, it comes up pink, we know it's gram-negative, and that can help us decide on how we proceed with our treatment options, right? Now, a lot of times we don't need to go to that step, right? A doctor can look at you and say, okay, you have this symptom, and this symptom. We're going to give you this medicine, okay? But if they say you have this symptom, and this symptom, and this symptom, and it seems like it's strep throat, but when they give you, an antibiotic and it doesn't work, then it may be something else. That it's mimicking the symptoms of strep throat, but it may be the result of a gram-negative bacteria. So some gram-positive and gram-negative bacterias, Staphylococcus, so like staph infection, Streptococcus, which causes strep throat, those guys are in the gram-positive group, right? Those are the ones that we're familiar with. These are the ones that we commonly see. They're responsible for, a lot of doctor visits, right? Strep throat, very contagious, can spread very quickly in classrooms and office buildings and things like that. Staph, even more so, staph infections. This is something that we really used to have a problem with before we had a really good system of cleanliness and disinfectant in hospitals. Staph infections ran rampant. You would go in with an open wound, they would put you in another room with a bunch of other people that had open wounds, and then soon enough everybody in the room died because they had staph infections. We were using the same kinds of instruments and that sort of thing, and we didn't have this really good disinfectant thing down like we do now, so we obviously know to separate those people and not allow that, because staph can spread very quickly, very rapidly. Gram-negative bacteria, a couple of examples of those would be E. coli. So E. coli can show up in contaminated food and that sort of stuff. These guys are going to be resistant to penicillin. And then also the bacteria Chlamydia trachomatis, which causes chlamydia. That's also a gram-negative bacteria. So again, a different form of treatment there. So whether they're gram-positive or whether they're gram-negative, all of that can be determined through a simple staining procedure, which helps us to identify bacteria and then decide what type of treatment we need to pursue. I'll see you next time.